Okay, students, in this section, we are going to see about proteasomes. What are proteasomes? Proteasome, they are a protein complex. Say, it is composed of so many protein subunits. And what is its duty? It degrade and recycle. Degrade and recycle what? Misfolded, unneeded proteins. So, such a type of protein complex is proteasome. Okay. Then it is present in all archaebacteria and eukaryotic organism. And also, if we take the case of a cell, we can locate proteasome in the nuclear compartment as well as the cytoplasmic compartment. So let's move to its structure. Have a look through its features, which are the important features of the proteasome. It is a large cylindrical multi-subunit protease complex. Protease means it can degrade protein. So that's why it's a protease complex, multi-subunit protease complex. It has a this portion, central 20S core is there. Then let's see the features of that core, 20S core. That portion, that core is conserved. If we examine archaebacteria till human, we can see that this 20S core is sequence, its amino acid sequence are conserved. There is no change. So that is one important feature, and its shape is barrel shaped and is composed of four annular rings of protein subunits. So this from this red to here, these are the four rings. 1, 2, 3, 4. So that 20S core is composed of four annular rings of protein subunits. In each ring, we can see so many proteins. Okay, then this end one, this red one, at both end we can see one red ring. Those red rings are called alpha rings, and it is sandwiching to beta ring, that blue color. Beta ring, I hope it's clear. Red one is alpha, and the middle one is a beta, beta ring. Okay. Okay, that beta ring, that's the important portion. It inside that region contains so many proteolytic enzymes. So that enzyme is performing the degradation duty of unwanted proteins. And this alpha ring doesn't have any enzymatic activity, no enzymatic activity. Then the both and we can see some cap-like structure. That cap-like structure, we can call it as 9DS caps. They are the regulatory units. Regulatory units means what is its function? We can understand. It is select. That's the function. It is select the protein to be admitted inside this proteasome, that is selected by this 19S caps. Okay, and also as we look to this cap, some proteins of this cap have ATPase activity. ATPase activity means what? It can hydrolyze one ATP molecule and it can release energy, and using this energy it can unfold polypeptide chain to facilitate to make that degradation work easier. So that is another importance of that cap. It select as well as it unfold the polypeptide chain using its ATPase activity of some of its protein subunits. So let's go and see how that degradation, how the proteasome degrade the unwanted proteins. It is an ubiquitin mediated pathway. Ubiquitin is a protein, and this is an unwanted polypeptide chain, or we can call it as a protein. To one of its end, so many ubiquitin molecules are added. So then we can call this protein as ubiquitin tagged protein, and that process is called ubiquitination. Okay, then there present our machine. This is a proteasome machine, and as this protein got tagged with the ubiquitin, it can move towards that proteasome, and looking to that tag, this cap select. Understand, this is the protein need to be degraded, and first the cap what it is doing is that it removes that tag. So all the ubiquitin proteins are removed by the cap proteins, and it is an ATP dependent process. We can call this process as deubiquitination. Okay, and this removed released ubiquitin again recycled to tag another protein to be degraded. So, after removing the tag, this protein will enter, or polypeptide chain is entering inside this 20S core, and inside we have learned that we know that inside this present so many proteolytic enzyme, and it degrade that polypeptide chain. As it undergoes degradation, we know what will release, is possible to release amino acids, and this amino acid again can used up by the cell for the synthesis of another different type of proteins. Okay, so that is a process that's about proteasome. Is so simple, and it's very important. It's a very important or means structure. It's helping the cells to remove unwanted proteins. Okay, thank you.
I'm an icon, man. And my girl about a 10. I should fuck her in a Benz.